Hello children, I welcome you all to the sixth session of the chapter Cell in Biology. Before I proceed to today's topics, I would like to have a quick recap of the last class topics. In the last class, we have finished the nucleus part, the brain of the cell. We have done the internal part of the nucleus about the chromatin material thickening, formation of the chromosome, and what is the function of the chromosome, how chromosome formed and why it is formed. That also we have done. Then we end up our session with the prokaryotic and eukaryotic cell. We discovered that a prokaryotic cell is a primitive cell in which there is no membrane, nuclear membrane. The nucleus is a just rough cloudy structure, and because it is not having any nuclear membrane, that is why there is no membrane bound organelles are also found in the cell. So there is a question arises that if these cells do not have the membrane bound organelles, if we take an example of the bacteria which are called as a photosynthetic bacteria, which does the preparation of the food with the process of photosynthesis, then how? Because plastids are not available. So how they do? They have some other structures, bag-like structures, which is doing the function of the chloroplast in them. Okay, now before I go to the next topic, I would like to tell you that today we will begin with the third important part of the cell, that is the cytoplasm. Cytoplasm is the internally filled, which fills the cell internally, in which all the other parts are floating. But the cytoplasm is not present inside the nucleus. Inside the nucleus, what is present? The nucleoplasm is present. So you can see the animal cell or eukaryotic cell in which there are so many cell organelles are present. You can see that each one of it, we will discuss it in detail one by one. So this is the nucleus, and inside the nucleus this is the nucleolus. These are the granular ribosomes. These are the centrioles which helps in the formation, which helps the cell in the time of cell division. Mitochondria, which is the energy storage area. Then you have the Golgi apparatus, which is helpful in the formation of the lysosomes. Okay, endoplasmic reticulum helps in the formation of protein and the lipids. Plasma membrane is the outer covering of the cell. Okay, now we begin with the cytoplasm. Cytoplasm is what? Cytoplasm is having the content of the cell, is carrying the content of the cell except the nucleus. It has two parts, cytosol and organelles. Okay, so the cytosol means the fluid part of the cytoplasm, which is homogeneous in nature. What is the meaning of homogeneous? That means uniform at all. And crystallocolloidal liquid that contains substances, water, ions, enzymes, vitamins, amino acids, carbohydrates. So many things are present in the cytosol. Okay, and then you have cell organelles. Cell organelles are what? The sub-microscopic structure which has its own structure, which has its own special function for the cell. Okay, but the cell organelles may be bounded by a membrane and may not be bounded by the membrane. In eukaryotes, all the organelles are membrane bound. In the prokaryotes, they don't have the membrane. So here we come to the last line. Virus lacks cytoplasm. Why? Because virus do not have a cell membrane. So there is no cytoplasm present in the virus. So what happens? Because the virus is not having any membrane, so that is why it does not have the life without entering into host body. When it enters in the host body, then only it gets all the nutrients, and then it gets its existence, or its you can say the identity, and then there itself it starts multiplying. So okay, and you know that whenever we make a slide to see the cell structures, we see that the cytoplasm part only takes very less stain. What is mostly taken areas? The stain is taken by the outlines of the membranes of the organelles, the nucleus, that part. So it shows the organelle part of the cells. Okay. Now, before I go to the organelles, I would like to give you the brief of the cell organelles. What is the meaning of the cell organelle? Cell organelles means they are every cell has a different types of membranous structure, and each one has its own function and structure, and they are different to each other. They are visible and it has its own structure and function in the cell. So first one, we will start with the endoplasmic reticulum. You have seen that endoplasmic reticulum starts where the nuclear membrane ends. Just it is like in the form of the folds. Some of them are having the circular structure, some are in the tubular structure. So endoplasmic reticulum is a network of interconnecting membranes distributed throughout the cytoplasm. It starts from the nuclear membrane and ends up in the plasma membrane. Okay, so the internal compartment, the area which is filled inside the folds, is called as the lumen, and it is a separate part of the cell with a distinct protein and ion composition. That means it carries a separate protein and ion. The foldings of the ER generate surface area much greater than that of the plasma membrane. So it helps in the process of digestion, grasping the things, sending the things out, everything. At the site, certain sites, you will see that ER is continuous with the nuclear outer envelope. That means I said it begins from the nuclear membrane and ends up in the plasma membrane. There are two types of ER. One is called as SER, one is called as RER, because where the area near the nuclear membrane, the edges, the surface of the endoplasmic reticulum is studded with the ribosomes, the granular structure. That granular structure helps in the protein synthesis in that ER. Okay, that helps in the formation of protein. So those part of endoplasmic reticulum which has surface with the ribosomes, they are called as rough endoplasmic reticulum, and the other one which does not have the ribosomes, they are called as smooth endoplasmic reticulum. Definitely, if ribosomes are present, they will have a certain specific function, and if it is not there, it will also have a specific function. Okay, the main function we will discuss later. Now, you can see that we are talking about the rough and the smooth. You can see that this area which is closer to the nucleus has dots. You can see that this is the rough, this is the smooth which does not have a dots. And this is the lumen, the internal part is called as the lumen area of the endoplasmic reticulum. You can see the structure here. Some are looking like a tubule, some are looking like a vesicles, some are looking like a oblong structures. So the ER is also present in the three structures, the tubules, oblong and vesicles. Now, as I told you that the area which is doing the manufacture of the protein are the rough endoplasmic reticulum, and the area without the ribosomes are having the function of lipid synthesis. And these lipids and proteins helps in number of ways. Some of them acts in the formation of enzymes and hormones for the cell. Some of them helps in the formation of the cell membrane. So those protein and lipids, when they form the synthesis of the cell membrane, that process is called as biogenesis. What is it? Biogenesis is a chemical process in which the proteins and the lipids formed by the endoplasmic reticulum helps in building the outer membrane. Okay, along with these functions, the ER is also responsible in the transport system. It transports the protein formed into the neighboring cells or to one part or the other part of the cell for their requirement. Now, here we have jotted down the function of the ER. Rough endoplasmic reticulum is the site of protein synthesis. Why? Because ribosomes are there, and ribosomes are the area where the proteins are formed, and SER forms the lipid. Proteins and lipids which are formed on the ER are used to make the cell membrane, and this process is called as biogenesis. It also helps in the transport of the things, proteins, lipids and other things, in the intercellular and intracellular. That means from cell to cell, and within the cell also it does the transportation support. It gives the because it is making the framework in the cell, and it is starting from the nucleus and end up in the plasma membrane. So it also gives support to the cytoplasm. Now, not only this, in the animals, the SER which is present in the liver cells, they helps in detoxifying the poisons and the drugs. Okay, so these are the main functions of the endoplasmic reticulum. To differentiate between the both, we have ribosomes are absent, ribosomes are present. They form lipids and steroids. They synthesize protein. They are mainly formed of vesicles and tubules, but they are formed of cistern, the long tube-like structures. They are made up of that. Okay, so here we have the difference between the endoplasmic reticulum types, the smooth and the rough. Now we go to the Golgi apparatus. The next organelle we will do is the Golgi apparatus or complex. Now, they are also the Golgi apparatus. You can see that the structures are long, tubule type, then some are sacs type and some are vesicles, and they secrete some kind of chemical. Okay, what is the role of it? It uh, gets the synthesized material from the ER to it, and from here it transfers. And sometimes if something poisonous or some non-required things comes inside the cell, the vesicle portions of the cell of the Golgi apparatus forms the lysosome, and the lysosomes ingest those poisonous things and they digest and they do it exits from the cell. So this is the function. The Golgi apparatus is a single complex in the animal cell, and they are in the form of separate units in the plant, and there are many small units in the plant, and they are called as dicytosomes, dictyosomes, and membrane bound sacs of the Golgi are called as cystine. These are the cystains, and these circular parts are called as the sacs like, and which forms the lysosomes. Now, one thing more you should remember, the area or the part which is facing the nuclear membrane is called as a cis phase, and the area which is facing towards the periphery or the plasma membrane is called as the trans phase. Okay, the foldings are called as cystine. They occurs in the form of stacks, tubes, vesicles on their periphery. Towards the end, you can see the circular is towards the periphery, the tubules are towards the nuclear membrane. So this is the Golgi apparatus. And the Golgi apparatus was discovered by the scientist called as Camillo Golgi. Camillo Golgi. Now you can see that this is the endoplasmic reticulum, and this is the Golgi apparatus, and the portion which is facing towards the Golgi apparatus is called as the cis phase, and the face of the Golgi apparatus which is facing the plasma membrane is called as the trans phase. Now you can see that some of the vesicles of the Golgi of the endoplasmic reticulum where the protein was synthesized is transferred to the vesicles of the Golgi apparatus. It is transferred. From here, now suppose this is transferred from here, it is transferred to the periphery. Now, if something proteins for the use of outside, it is transported outside. Suppose if something gets inside, so these circular structure of the Golgi apparatus turns in the and forms the lysosomes. In that way, what happens? The lysosome ingests it and it secretes some kind of chemicals which helps in the damaging of those poisonous things, or they engulf or they kill them, and that is why the lysosomes are called as suicide bags. Okay, so lysosomes are formed from the Golgi apparatus. Now the function of the Golgi apparatus. The Golgi apparatus is involved in repair and synthesis of cell membrane. How? Because the protein which was synthesized by the endoplasmic reticulum, or the lipids which was synthesized by the endoplasmic reticulum, smooth one, they are transferred, they are packaged, dispatched and send it to the Golgi apparatus. The Golgi apparatus carry and send it to the plasma membrane, and if there is any kind of repair or synthesized has to be done, then they do that function. Now, second point is The lysosomes are also formed by the Golgi apparatus. Some of the Golgi apparatus, they turn into lysosomes, and the lysosomes kills those unwanted substances which enters inside the cell. All type of substances which are to be secreted or excreted are packed in vesicles by the Golgi apparatus for the passage to the outside. If something has to be secreted out of the cell or something has to be excreted out of the cell, it goes to the Golgi apparatus. Golgi apparatus turns into lysosome and then it does exocytosis. Okay, endocytosis is taking inside and exocytosis is done by the Golgi apparatus. Takes part in storage. Some things are stored like proteins and lipids are stored, modifying the cell membrane, packaging the various biochemical products by the different components of the cell. If something has been synthesized, they also stores in the Golgi apparatus. Components of the cell wall are synthesized by the Golgi apparatus. Even the plant cell, the components which is needed for the repair and the synthesis of the cell wall is also made by the Golgi apparatus. Special complex sugars are made by the Golgi apparatus. Some of the sugars are needed for the different function. It is also made by the Golgi apparatus. So these were the functions of the Golgi apparatus. Now, since we have done the Golgi apparatus, it is mandatory to do the lysosomes also. Now, children, whatever organelles we are doing, along with the function, their structures, you have to draw a rough diagram of it. I am showing individual diagrams to you. So, you are supposed to draw all the diagrams separately. Now, when you will head up the lysosome, you will draw the Golgi apparatus, and this is the lysosome which is formed. Okay, so this way you will draw. Okay, coming to the lysosome, you can see that this is the plasma membrane. Okay, now the vesicle portion of the Golgi apparatus turns into primary lysosome. The primary lysosome changes to secondary lysosome when there is some kind of food particles taken in by the cell by the phagocytosis. Phagocytosis is the process in which anything engulfs the things. That process is called as phagocytosis. Now these phagosome is connected with the primary lysosome and forms the secondary lysosome. What is the role of the lysosome? Lysosome digests the products. So when it digests, the useful things is taken by the cell and unuseful thing is sent out. That means the lysosome kills itself and it also gets destroyed, and the waste products are sent out to the plasma membrane by exocytosis. Lysosomes are also called as suicide bags. Why are they called as a suicide bags? Because they kill the unwanted things or the microbes or the bacteria or any unwanted components inside the cell by killing themselves also. That is why they are called as suicide bags of the cell. Lysosomes are generally found in animal cell and they lack in the plant cell. All small spherical vesicles covered by a single membrane contain digestive enzymes for the intracellular digestion and waste disposal. So remember, the spherical Golgi apparatus which is turned into lysosome, they contain some kind of enzymes which is helpful for the digestion. Intracellular, that means inside the cell, if anything has to be digested, if some of the organelle has become dysfunctional, that is of no use, so those things also being digested by the lysosome, as well as to expose out or to dispose of the things outside the membrane. These both the processes are done by the lysosome, and these enzymes which is doing this work in the lysosome is being synthesized by the rough endoplasmic reticulum. As I said, the enzymes, the hormones, all are what? A kind of a protein. And protein is synthesized by what? Protein is synthesized by rough endoplasmic reticulum. In damaged cell, as I said, if the cell is damaged in certain part, what happens? the lysosome burst some enzymes, release some enzymes, and they engulf the entire cell with its all components and digest it. So if the cell is damaged, the lysosome itself becomes bigger and it secretes some kind of digestive enzymes to digest the entire components of the cell. Now, what is the main function of it? This was about the lysosome. The main functions are destruction of foreign particles, intracellular digestion. Destruction of foreign particles means if anything has come inside the cell which is going to trouble the cell, that is digested by the lysosome. If anything has to be digested inside the cell, that also done by the lysosome. If any waste products are secreted or produced inside the cell, they are also being removed by the lysosome by the process of exocytosis. Now, time of metamorphosis. Metamorphosis means turning from old to the new. So if a cell is damaged, if that has to be repaired, that is also done by the lysosome. So the lysosomes are what? Lysosomes are the organs which helps in the digestion of the other organs of the cell. So this much I am going to do it today till the lysosomes. Today we have done endoplasmic reticulum, Golgi apparatus and lysosomes as the main cell organelles, as well as we have done the cytoplasm. This much for today, children. Hope you all understood things. Thank you. Have a nice day.